Good morning, all of you. Myself Subodh, and my topic of the presentation is DNA, peptide, and recombinant protein. So, DNA is a topic which we all have studied from our lower classes. DNA basically stands for deoxyribonucleic acid. It was established by James Watson and Francis Crick, and DNA is composed of two polynucleotide chains that coil around each other to form a double helical structure. So this chain of a double helix will contain the genetic information that is for the development, functioning, growth and reproduction of an organism and even viruses. So DNA will code for the traits that is made up of repeating units of nucleotides. Now what is nucleotide? You can see in this image that nucleotide, let me choose a color, that this is a nucleotide molecule which composed of three parts, that is phosphate group, pentose sugar and nitrogenous base. But if we remove the phosphate group, we only left with the pentose sugar and the nitrogenous base, and this combination of pentose sugar and nitrogenous base is known as nucleoside. Now this whole nucleotide molecule will be of adenine, thymine, guanine or cytosine. This is for the DNA. But if you talk about the RNA molecule, in place of thymine there is a presence of uracil. Now nitrogenous base are of two types, that is purines and pyrimidines. Purines are basically double ring structures and it is composed of adenine and guanine. Pyrimidines are cytosine, uracil and thymine. Base pairing always occur with purines and pyrimidines. That is known as the Chargaff rule. From this image we can get the information about the structure of the purines and the pyrimidines. So purines are double ring structure as I mentioned earlier, and pyrimidines are single ring structures. So according to a Chargaff rule, we can say that purines always pair with the pyrimidines. Exception is the RNA. Now adenine is always paired with the thymine, and guanine is always paired with the cytosine. But as in the case of RNA, we can see that adenine is paired with the uracil. Now this structure will give information about the pairing between the base pairs and the sugar phosphate backbone that is present in the DNA. So we can see that the backbone of the DNA is composed of sugar phosphate molecule, that is phosphate and sugar attached, and in the middle part of the DNA there is a base pairing between the pyrimidines and purines. So this base pairing is done by the pyrimidines and purines, that are adenine, thymine, guanine, cytosine, uracil, only uracil in case of RNA, and these are joined with the hydrogen bonds. So always adenine pair with the thymine with the two hydrogen bonds, and guanine pairs with the cytosine with three hydrogen bonds. Now from this we can infer that breaking of the bond of guanine and cytosine is more complex than adenine and thymine. So if in a DNA GC content is high, it means it cannot melt easily. So its melting point is higher than the other one which has low GC content. Now this is an image which will show about the diameter, length of the DNA molecule. So DNA composed of the two grooves, that is minor groove and major groove. This is a sugar phosphate backbone, and in between that we will have the base pairing of the nucleotides. Now we can see that distance between the two base pair is of 0.33 nanometer and diameter is 2.37 nanometer, and we can see that distance between us two major groove is known as 3.40 nanometer. If we count it in Armstrong, that is 340 Armstrong. Now we will talk about the central dogma. So central dogma is a process through which we can see that how the DNA molecule will change and lead to the formation of the protein molecule. So there are various processes where we can study here. So there is possibilities that DNA can make the DNA again, and DNA can make the RNA molecule. So if the DNA is making the DNA molecule is known as the self-duplication. It means it is duplicating itself, and this process is known as replication. If DNA is making DNA again, is known as replication, but if the DNA is making the RNA molecule, is known as transcription. So the enzyme used in DNA replication is known as DNA dependent DNA polymerase. That is, it is acting on the DNA molecule and again forming a DNA molecule. So DNA dependent DNA polymerase. But in case that it form the DNA from DNA, it form the RNA molecule from the DNA. So that enzyme is DNA dependent RNA polymerase. That is, it acting on the DNA molecule and forming RNA. This process is known as transcription. Now, if the reverse is happening, that is, the enzyme is acting on the RNA and forming DNA. Okay, so this is known as reverse transcription, and the enzyme used here is known as reverse transcriptase. So we can also name it as RNA dependent DNA polymerase. So all the reverse transcriptase enzymes are RNA dependent DNA polymerases. Now, if we form protein from the RNA molecule, it is known as translation. Okay, here here is the role of the enzyme tRNA, that is tRNA molecule that will help in the formation of protein. Now this will image of replication process, in which we can see that it is this self duplication. It takes place in the S phase of the cell cycle, and it will divide into two equal parts on the division. So replication is of three types, that is dispersive, conservative and semi-conservative. We will talk about this on the next slide. So we can see in the image that there is an old strand that is in blue color, and the new strand that is formed from this old strand is of purple color. So every strand, each strand of the DNA molecule will form a new strand and duplicate itself. This is the process known as the replication. So in this image we can see the type of replication, that is semi-conservative, conservative and dispersive. So in the semi-conservative model, we can see that DNA is semi-conservative, as one strand of parent DNA and the other strand is of the daughter DNA, and it will replicate itself and form a complete new strand, and a strand with half daughter DNA and half parental DNA. But in conservative model, whole parental strand is conserved, and it will form a separate daughter DNA and separate parental DNA, which will replicate further. Now in dispersive model, we can see that the parental strand is present in short stretches in the daughter strand. It means it neither completely get attached nor it will semi-conservatively attach, but it is attached in small stretches, and in the next generation it will present only in the small amount of in the daughter DNA. Now we will talk about the transcription process. In the transcription process, we will form the RNA from the DNA template strand, and only one strand is participate in this process. But we can see in the replication that there is both strand is replicating on its own. So enzyme involved in the transcription process is known as RNA polymerases. In translation, we will form the protein, and the and the tRNA is govern this whole process. We can see the structure of the tRNA molecule here, where we can see there is a T loop, D loop and anticodon loop. T loop is also known as pseudouridine loop, and there is an attachment site where of 3' end, and there is a presence of a conserved sequence of CCA. Now we can see that in the transcription we will have to form the mRNA from the DNA molecule. So only one strand will participate in, and the formation of mRNA will take place, and from this mRNA transcript we will form the polypeptide chain that will act as a protein molecule. Now we will talk about the peptides. So peptides are the short chain of the amino acid that are linked together by a peptide bond. So the peptides are of two types, that is oligopeptide and polypeptide. So oligopeptides are the chains of amino acid which are less than 15. If they are less than 15 in number, we can say that these are the oligopeptides. But if these are more than 15, then we will consider it in the polypeptides. So there are three types of peptides, means oligopeptides, that is di, tri and tetra. So these are the naturally occurring biological molecules which are found in the all organisms. So we can see in this image about the peptide bond formation that two amino acid are involved in formation of a one peptide bond. That's from this removal of water molecule will take place and a peptide bond is formed. The bond C double bond ON is known as a peptide bond. Now we will talk about the polypeptides. So polypeptides are long, continuous, unbranched chains of amino acids that are more than 15 or 50 amino acids. These are comes under the biological polymers. So these are the biological polymers, or we can say that oligomers of nucleic acids, and these are oligosaccharides or polysaccharides, we can give that in the name. So polypeptide contains more than 50 amino acids is called a protein molecule. If more than 50 amino acids are joined together, it will form a protein molecule and it will arrange as a coenzyme or cofactor, or it can also form the macromolecules like DNA or RNA. Now we can see from this image that a protein molecule will undergo a small will have a different unit, that is polypeptide chain will join together to form a protein. But if we break the polypeptide chain, we will get tripeptides, dipeptides and amino acids. So amino acids are the long, these are the, if we make a long chain of amino acid, we can form the protein molecule. So we can see that if we break the protein molecule, we will have the polypeptide chains, and if we break the polypeptide chains, we will have tripeptides or dipeptide. From that we can get the amino acids. Now now we will talk about the recombinant proteins. So recombinant protein is a protein that is coded by the manipulated genes or recombinant genes. What we will mean about the manipulated genes is that we will modify some genes by the help of RDT, that is recombinant DNA technology, so that we can make a recombinant protein that will work according ourselves, that what we want from that gene. These are generated by specific recombinant expression system. We will use the expression system for their specific recombination. Recombinant genes is a new genetic combination of the gene that are manipulated by insertion or deletion. So we can insert any sequence or we can delete any sequence for the modification of a gene so that we can get the desired product. We will use the RTT, that is recombinant DNA technology, for this for their formation. So this is a diagrammatic view how we will form the recombinant protein. So we will have the donor DNA, we will cut it at the specific position with the restriction enzymes, and then we will have the DNA ligase so that we can join these ends. Now we will introduce these into the bacterial cells, and these are divided in the bacterial cells, and we will have the recombinant DNA molecule that will replicate inside that bacteria. We can get the protein from that cells, and we can culture more number of bacteria. So this is the transformation process of a cell. We will introduce the foreign DNA inside the cells or with our gene of interest, and we will select and expansion of that positive clones. So we can expand them, and isolation of plasmid expression vector. In that we will have our desired gene, and we will introduce in the bacterial cells, and we will get the protein of our interest. So that's all about the presentation. Thank you.